Thanks, Alex. Actually, I'd say it started almost three and a half years ago. And actually, so the work I'm going to talk about is in two papers, one of which came out in January 2016, and the other one was just accepted after 5 revisions of hell. All right. And actually, I'm about to start a new chapter in my life in academia in January, after more than a decade at Microsoft Research. I can't tell you where just yet, but hopefully very soon.
I don't know if I was allowed to say that.
Oh, you are allowed to say that. Just not where, because yes. So I think I probably don't need to explain the intro to CRISPR, but I feel like I should, just to make sure I cover the basis. So I'm sure everybody here knows what gene editing is, and that there's a long history, and that it's basically like cutting and pasting and these kinds of things. Right. And so, a few years ago, this kind of thing was very, very expensive and very, very laborious. And it wasn't until 2012 that CRISPR came to make these things much cheaper and much faster and more scalable. And now people are doing all kinds of things with CRISPR. But there's two sort of core tasks that people started to do with CRISPR and probably are the main functions of this technology right now. And so one is sort of a classical edit where you change an A to a T. And the other one is gene knockout. And because all of this work is joint work with John Dench and many other people, but John Dench is in the screening platform and he is very interested in knockout. And so the focus of all of this work is knockout. But of course, the key task to both of these, the key thing to both of these things is precisely cutting the DNA. And so the modeling we've done has, for the most part, focused on that. And so it stands to reason that much of what we've done is applicable for this first task, but we haven't really established that right now. This CRISPR has sort of slowly made its way from, first probably before the top tier journals, other journals, and then slowly into the popular media. New York Times, CNN, and then who saw this cover piece on Wired from a few years ago? This is everything that's written on this cover is actually in reference to CRISPR. So apparently CRISPR means no hunger, no pollution, no disease, and the end of life as we know it. It's the Genesis engine. So that's, I guess that speaks to the hype that is a little bit around CRISPR at the moment. And so there's also some backlash as we see. This is now pretty old. I haven't followed it. I don't know if it's new, but that was a direct causal result of the cover Wired magazine. So, but the reality, as I'm sure everybody knows here, is that there are already a lot of very promising proof of principle for therapeutic applications, and it's already routinely used in the lab for drug screens and all kinds of other things. So it is hyped, but it's also obviously very, very useful at the same time. And so I'd say there are two main problems, and I'm going to sort of abstract away what a biologist would say to a computer scientist's mind. So in my mind, there's two main problems, which are the on-target and the off-target effects. And so again, I'm going to focus this talk on gene knockouts. So remember, the goal is that for some gene, let's say this one right here, I want to prevent it from functioning. And so the way CRISPR works to do that is you basically throw in these scissors, which are the Cas9, for example, and it's going to make a cut to the double-stranded DNA. And then it will repair itself. If I don't give it a template, it'll just kick in an innate repair mechanism. It will try to repair itself, but with some reasonable probability that repair will fail, and that's what will cause the gene to not function anymore. And so CRISPR has a few constraints of where you can apply these scissors, and that's determined by Cas9 or something like Cas9, which does the cutting. And so Cas9, for example, needs on one of the strands to have a GG, and that's the PAM site. And so basically it recognizes a PAM site. You can imagine GG is very promiscuous, and so you can cut in many, many places. And so now imagine, like here I've just shown 4 places that I could in principle cut, but there's actually hundreds of places for a single gene. And it turns out that some of those work well to knock out the gene, and some of them don't. And in some genes, it might be that many of them work well, and in other genes, it may be that very few of them work well. And so already, like now, day to day, people are trying to do these gene knockouts, and they're not sure which thing to apply. All they know is that Cas9 needs a GG there, but they don't know like which of these. And so this is, as you can imagine, we can use machine learning to improve this kind of task. And the flip side is the off-target effect. So now imagine I've selected this particular site in this gene, but I have the whole rest of the genome. What I haven't explained yet is it needs that GG. Yes, Yossi? What's that?
So when you say some of the sites failed to knock out the gene, there's kind of two ways that I can see that happening. One is that the scissors don't work, that they don't cut.
Yeah.
And the other option is that they do cut, but then cutting does not.
That's exactly right.
So is that, are you putting those two together, or do you
So
need one or the
people
other?
do different things in the data we have, which I'll go into some detail about. The supervised signal is going to be the result of an assay, and that assay is a functional assay, which tells you the gene's not functioning. So it requires both the cut and the lack of function, but in the off-target stuff, we sometimes model just the cutting, so it kind of depends what's available to us as data people. And from what we can see, often these are highly correlated in that a model trained on one works well on another, and I'll get into a little bit about that later. Yeah. And so for the off-target, though, beyond the NGG, as I'm going to explain, there's a guide RNA that also needs to match, like where you design. You say you want it to go here, then you need to have an NGG here, but then you also design something that's complementary to that, to the DNA there. And it may be somewhat complementary also elsewhere in the genome, and it may have the capacity to also cut there and potentially disrupt. And so these are the off-target effects. And obviously, we want to ideally eliminate these, or, more practically speaking, reduce them, and also maybe understand where they occur, because maybe some are worse than others. So these are the two problems that I'm going to talk about. Are they clear to everyone? Okay. So people tackle this, obviously, in the laboratory, improving the molecular biology and all kinds of things like that I'm not going to talk about. I'm a machine learning person, so I'm going to tell you how we can use machine learning to also help us for these problems. So I usually give probably everybody at the Broad knows this history, that it came from a yogurt company, was the one who figured out what CRISPR actually does. Does everyone here, who put up your hand if you already know the company? Oh, okay, very few. So, okay, super interesting, right? So for decades, people, sort of poorly funded biologists looking at bacteria and things like this were studying CRISPR, but no one knew what it was, and that's why it was sort of obscure, low impact, and poorly funded. And Danone, a yogurt company, which obviously has, and researchers at that company, obviously, discovered what it was, and they did because it had a real impact on their bottom line. And the impact was that they discovered it was associated with a naturally occurring immune defense mechanism in bacteria, and that had practical consequences for them because some yogurts in their vats were being infected, and others weren't. And so they had to throw out the ones that were infected, basically, and so they would pursue this, and they're the ones who actually discovered that CRISPR is part of this bacterial immune defense mechanism, and I'll explain that in just a second. And then you can see a pretty short time later, and of course there's much other work that led up to these developments, but in 2012, people realized how to take the core machinery of this natural defense mechanism and use it for general purpose gene editing. And of course, that has many implications for all kinds of things that humanity cares about. So now let me go to the system that Danone had figured, or the researchers at Danone had figured out. And so this is a bacteria. And if it gets infected by a virus, this virus has this RNA here. And if the bacteria survives, what it will do, or what it can do, is it can cut out part of the viral RNA, and it can put it in its own DNA down here. And it sort of takes a snapshot. So you can think of that as like a viral memory that it's storing in its own DNA. And then if it gets infected again, it's now kind of primed because it now has the capacity to recognize these things very quickly, much as yours and my adaptive immune response can be primed by a vaccine. Actually, I just had a vaccine this morning to recognize things very quickly. And it has innate these sort of scissors in the system which do the cutting. And so these scissors are kind of, floating around, and they are primed to recognize. And the next time it gets infected, it will cut it. And it therefore has the potential to fight off this virus. So that's how it's a naturally occurring immune mechanism. But as you can imagine, there's something here where you have a precise sort of thing that guides it to a particular piece of RNA, and it makes a cut. And so, of course, from everything I've just told you, it's no surprise that people, or maybe it is a big surprise in retrospect, it's no surprise that people figured out how to use this for gene editing. And so right now, the system is, in fact, you can sort of take these bits and pieces and put them in almost any organism, and they will still function. And so what I used to call a sort of viral memory, and one thing I forgot to mention is how how does the bacteria sort of know that this region here is storing these memories, and how does it delineate the endpoints? So what it does is it puts little patterns here. Does anyone know what these patterns are? These are the clustered regularly interspaced palindromic repeats. So that's actually where the name comes from. So they're sort of the delineation of these things in the native system. Right, and so, but now what we can do is we can say, let's take some, like maybe this is a human genome here, and I know I want to either edit it or disable in this region of the genome. So what I do is instead of like waiting for bacteria to be infected and to find part of a virus, what I do is I just synthetically generate this thing down here, and we call that's the guide RNA, and we make it complementary to the region in the genome that we want to cut. And again, we have the constraint that needs to be, if this is Cas9, there needs to also be a GG here. Otherwise, even with the complementarity, the Cas9 just won't work. It won't go in there and it won't do its thing. Right, so does people understand how the basic system works? Is there's a guide RNA, it's about 20 to 30 nucleotides long, and you throw it in with some scissors, and then it goes to the part of the gene. In an ideal world, it goes to exactly where you want and cuts only there and does what you want. Of course, it's not an ideal world. And so, right, so now I'm going to tell you how we use machine learning to tackle the two problems I told you about in the beginning. So one is how do we get better on-target efficiency using machine learning? And we've dubbed this model Azimuth. And then afterwards, I'll tell you how we've reduced the off-target effects, which we call elevation. I don't know. Oh, so this is joint work with Nicolo, who was speaking here last week. And I think he liked these names. That's how these names came to be. Right. So what's the basic setup? This is not actually super fancy machine learning, I would say. The off-target problem is much harder. The on-target problem is a little bit straightforward, but just the due diligence we did, we spent months and months making sure that we were doing things really robustly and solidly. And so, in any case, the basic setup is as follows. It's a supervised machine learning problem, which means we need some inputs and we need some examples of how well those inputs worked. And then we're going to try to build a model f of x, which captures the generalities of this input-output and then can be applied to new examples. So that's just the basic setup of supervised machine learning. And so the RNA guide here is going to be, in its original form, will be like, we use 30 nucleotides. And then we have some measure that I'll explain that tells you how effective it was at knocking out the gene or not. Right. And so, again, this is, I think in this seminar series, probably everybody here knows this kind of thing by now, but, oh, Yakir?
I was just wondering, in our mind, should we be thinking about one fixed gene right now?
No.
No.
Well, you can, but you need not. So the training data that I'll talk about has about 15 genes in it. So yeah, so this can be, yeah, no, you can think of this as anywhere you want, and what we want is we want as much training data like this as possible. And if we have more than one gene, we're going to have a much better training data set. Yeah. There are issues that I might go into a little bit about dealing with the assay measurements and how they may or may not be consistent across different genes and how you deal with that, but that's not a machine learning problem. That's like an assay issue.
I see, but we should be imagining like one F that is a function of
yes.
30 base pairs.
Yes. Yes. Yeah. Sorry. Yes. I didn't understand. That was your exact question. It's any guide and any amount of effectiveness. That's right. But keep in mind that people design so the training data is with guides that were designed with the hopes they would work, and so people don't design promiscuous guides, for example. So in general, these guides don't have a perfect match anywhere else in the genome. So it's not like we're picking random guides in the training data, and I think that might make it a bit weird. Yeah. Right. So we have these, we're going to have to somehow take that 30 nucleotide sequence and convert it into features. And we're going to need some measure of effectiveness, and then we're going to have to pick some sort of model class. And I mean, these are just the core components of anything in machine learning. And so, I'd say, luckily, we have some very smart collaborators at the Broad who know how to do this. So what this is how did we get the supervised data? So that's how did we get these labels over here for how effective each example was? And so we did it John and these guys at the Broad did it in two ways. So in the first example of data we had, this one I think was 7 or 8, they're each about 7 or 8 genes for a total of 15 genes. And flow cytometry was only cell surface proteins. So proteins that are expressed on the surface of the cell, because they're expressed on the surface of the cell, you can attach fluorescence markers and you can do sorting on them and things like this. And so because of that ability, you can then, using ingenuity of biologists, tease apart which ones got knocked out versus which ones did not get knocked out. And back to Yossi's question, this is not cutting, this is actually like a functional, is that protein expressed functionally or not? Such that it actually gets tagged with the fluorescence and you can see it. And the other assay is, and keep in mind, that's very specific to just cell surface proteins, so you can't use that in general. And the other one is also very specific to a different class of genes. Those are genes for which it's known that they have a particular kind of drug resistance. And so I'm not gonna go into the names of the drugs and all these things, but the concept here is that it's known that for some genes, when you apply a drug, that if that gene is knocked out, it will survive, but not otherwise. Sorry, to a cell line. Apply it to the cell line. If the gene has been knocked out, the cell will survive, but not otherwise. So again, just from this prior knowledge of the drug sensitivity, you can do a readout based on did the cell survive or not, and get a sense of how much it was knocked out. And so I'm emphasizing that these are very specialized because for a couple of reasons. So one is, why are we using machine learning? If you can just measure these things in a lab, well, I think as most people here know, yeah, we can measure them in a lab, but only for this is months and months of work to get this for 15 genes and a lot of money. And of course, we want to be able to do this genome-wide. And so it's a scale problem, as we can't easily do this for the whole genome. And more importantly, as I don't know all the developments, but you can see these are very specialized. So even if we had the time and money, you can't even apply these assays genome-wide anyway. So that's how we measure it. And then the last, so now let me go a little bit into the featureization. So there's a rich history in computational biology with machine learning on how to go from nucleotides to numeric values. A lot of it came from the support vector machine community, which actually when I was a grad student, they were as hyped up as deep neural networks are right now. And so you can, so in support vector machines, I guess the nuances here aren't important, but they build kernels which have implicit feature spaces, and you can back out of those and just get explicit feature spaces, which is what we did. So the idea is, do I have a slide? Yeah, so just a simple example would be, say we want to know just, is there an A in position 1 or a T in position 1 or a C, which letter is in position 1, then we can basically convert each letter. This is just a standard one-hot encoding. So there's 4 letters, so each letter gets 4 digits, and one of them is on. And so now you can just encode in the first position, this one had a T, and the second position is the next 4 digits, it's had a G, and so on. So you get this really long string of 0 and 1, which is now suitable for any machine learning model that you'd like to apply. And you can also do this for dinucleotides, and you can, this, as I just showed you, is I'm concatenating them according to their position, but you can also do things like count how many there are, and you can do interactions of them, and things like this. So this is sort of one of the main features that provides a lot of the predictive power. And so you can actually see here, so some people, people had been doing a little bit of machine learning before we started this, and they were using those features and some of those features. But you can see, if you look at the feature importances here, those ones that tell you the position-dependent nucleotide, either order 2 means 2 nucleotides or order 1, these come out as actually the most important thing for this problem. And you can see that when you add these other kinds of features that hadn't been there, I guess actually those are the starred ones, right? People hadn't been using those, then you boost the performance up on these various data sets. So FC is that flow cytometry, res is the drug resistance, and this is their combination. And so one thing that was kind of neat is John had, and I guess other people had this hunch that the thermodynamics should play a role. And you can see in this video actually how Cas9 comes in and why that might be. So Cas9, what it does is it goes in and it actually has to find the PAM, and then it has to pull apart the double-stranded DNA before it makes a cut. And because it has to pull it apart, that obviously requires energy, and the energy it requires has to do with the thermodynamics of the bonds there. And so if you can model that, it's to the reason that we could do better, and in fact you can. And we did a few things. So John was aware of a crystallography paper that looked at more precisely how things were happening, and said, I think if you break it, we did the thermodynamics of the whole guide, we also did it of parts of the guide according to those crystallography experiments, and that also seemed to be useful. Right, and so now in terms of modeling, again, like, so this is just standard off-the-shelf stuff here. We used, yeah.
So what does it mean to turn, like, is the thermodynamics, you have some function that goes from the sequence to some notion of how much energy is required to separate?
That's right, yeah, and we didn't develop that. People have developed these things and made the software available. Yeah.
So you included as a feature some funny nonlinear function that someone else has learned.
Yes, exactly.
That it would be hard for the thing to learn by itself.
Well, right, so a lot of, I mean, that's interesting because in the modern era, people are like, why do you need features? Just throw it in a deep neural network, right? So we don't have anywhere near the amount of data. Yeah, I know you know that, but just to be clear for everyone that in principle, you could probably learn this if you had enough data, but we're like, we tried deep neural networks just for kicks, and, if we'd been really like attention seeking, we would have just used them. They work just as well, and then it would have been on the front cover of Science saying deep learning plus CRISPR, right? Which, has probably come since, and I get asked to review these kinds of things, and they're usually crap and not needed. But I did not mention any names. I know this is recorded.
You can't do it because there's not enough data.
At the time we did this, I think that it's starting to change, and there may be enough data in some domains. So there's many flavors of the CRISPR system also and things like this. So I have seen some work that looks promising that's not public yet. So it's not all crap, but there's certainly a large attempt by everybody to use deep learning for whatever without doing due diligence that it's actually needed. And so in this case, it didn't help any.
I imagine that must go hand in hand in those cases with trying to design more high throughput assays for.
Yes, that's right, exactly. Yeah. Mm-hmm. Right, and so now I've explained how we have for 15 genes, we get these measurements of how the knockout works. So you get some sort of efficiency, like it's not just binary, it's sort of on average, this is how much it knocked things out. And we're gonna use regression trees, which probably after 40 of these MIA talks, someone has explained regression trees, but if not, they're sort of a very nice model in the sense that they can be highly nonlinear, and you can control the complexity well enough pretty easily, such that even if you don't have a lot of data, you can still use them. And so you basically it picks one feature at a time and decides to split on it. If it's a real value, it figures out where to split. If it's a binary or categorical, it just chooses that. And it basically, in a greedy way, kind of constructs a tree like this, which has the effect of, if you had only two features, of sort of partitioning the space. And then within each part of the space, it now can make like a regression prediction. That could just be the average of the examples that fall on the leaf there, or it could be building like a simple linear regression model with those examples that fall into that leaf. Question?
The negative examples, or if there are negative examples, or?
So negative examples would be only if we were doing classification. So I'd say one thing is that everybody in this field likes to do classification, even though all the assays are real valued and they're throwing out a ton of information. And we've shown that in two of our publications, that you throw out a lot of information by doing that. So if you don't do, like in classification, there's this notion, but in regression, it's a real valued number. So there's the low ones work poorly, the high ones work well. Or maybe it's the other way around. I don't know. Each assay is different, but.
The good candidates to begin with in this.
So there is a bias in the training data set in that we're not sampling randomly at uniform from the input space. Those are designed by people who say, I know I want it to be unique, like not match elsewhere and things like this. Yeah. That's right. And so, so that's how a regression tree works, and we actually use boosted regression trees. And so again, I don't know if people have gone over boosting in this, but boosting's really cool. There's a very solid theoretical foundation behind it, which I won't go into, but there's also a very nice intuition. And so what boosting does is it takes like some sort of pretty weak model. In our case, we're gonna sort of force the regression trees to be pretty simple by keeping them very short, mostly speaking. So if you keep them short, it can't use very many features and it can't really overfit. And so when you have these sort of very low capacity models, we call these weak learners. And you could use a different kind of weak learner, anything that's just sort of not a super rich model. And what boosting is intended to do is to start with a weak keep relearning on a slightly tweaked version of the training data set. each time it reweights the examples. It effectively reweights them and learns another model. And at the end, what you have is you have a whole bunch, a whole ensemble of this weak model. In this case, the weak model is the regression tree. And then it gives you a rule on how to combine them. So that's sort of the abstract concept. And actually, to give a bit more intuition, what it actually does is, what you do is you take your original training data set, and you fit, in this case, the regression tree, like one that you forced to be pretty small, and then you look at how well it did on each example, in sample, not out of sample, so the whole training data. And then what you do is, for those examples that it did badly on, you upweight them proportional to how badly it did, and then you learn the next one. And so that's how you keep, and you can keep going, and then you weight each of those models by how well it did overall on the accuracy on that training data set. And so, I mean, I think there's some intuition here that sounds very pleasing, but as I said, like, if you go and read about this, there's a whole set of theoretical reasons that this is also very sensible. And there's interpretations of this model also in different ways that are quite interesting, but I won't go into here. I just want to at least give you that intuition, and that in general, this can be a very nice way to get a very powerful model, and it turns out that it was for us the best model. And so actually what you can see here is on these data sets, so when it says train on FC, test on FC, this is obviously with cross-validation. We're never testing on what we trained. Actually, in all of these, it's cross-validation. And in particular, the flavor of cross-validation that we do is, so right, cross-validation, you normally like chunk up your data into, say, 10 folds. So you hold one fold out, train on the remaining 9, test on the last one. And here we're actually going to make sure we do it by gene, meaning that we're going to test one gene, will be one holdout, and we're going to train on the other ones. And the reason we want specifically to do it that way is it sort of mimics the use case in the real world, right? Like, chances are people are not going to be using any of the genes we had in our training data. They're going to be using some totally different gene that we had no data on. And so it's sort of the closest we can get to the use case. And if you do it the other way, you actually can get a higher accuracy, because sometimes, I don't know if we quantified that or not. But anyway, so John and his colleagues had just actually published a paper when I met him, or we're in the middle of revision, and this was their model, and it was the best. It was the state of the art at the time, and it was a combination of SVM and logistic regression, and that's the performance it has. And then the model that we published, and the models, all the other models are actually just our own models that we tried out of curiosity, so they're shaded a bit lighter because they're not sort of competing models. And you can see that the boosted regression tree is what did well, but you can see that actually super simple models do well here, and that's just because, one, like, there's very little data, and probably the other reason is often, I think, especially in biology, that linear additive signal is very powerful. And so you can get away with that. And so, so even though this is kind of simple in machine learning, as I said, we spend months and months sort of really trying to assure ourselves that this was the best thing, and we didn't overfit, because we knew if we did a good job, people would really use this. And it was really satisfying the year after this came out that the two different individual or sets of people actually showed this was the state of the art on data we'd never seen. And so a lot of people did start using this tool that we put out. And so, right, in fact, it's actually been adopted by 2 startups. And it gets used all the time. It's available through the Broad website. It's also, the source code's available.
Why do you use Pearson correlation instead of square? Like fitting you in position?
We don't use Pearson correlation. We use Spearman correlation.
Sorry,
and the reason we use, so I glossed over that, and I'll go into a bit more detail with the off-target, but there's a real problem with the assay readout is not super comparable between, say, like, different genes, let alone the different assays. And so what we do, and I've glossed over it here, is we follow what John did, which is to rank normalize within a gene. And so when Kira was saying, is there anything gene specific here? That's the only sense in which something is gene specific. And it's not quite the right thing to do, because what that means is that every gene now has the same scale. But the reality is one gene could have many things that work and another gene might not. So this is not ideal. And if there's time at the end of this talk, I'll tell you we actually have like a separate machine learning project we've been working on in parallel to address this, but it's just like we didn't have time to do it with everything else here. Yeah.
Do you independently assess that your predictions are unbiased?
We don't care about biasness. You care about if you care about precise values, right? We care. So let me. I didn't answer I got sidetracked with my own answer. So the more direct answer to your first question is we do Spearman correlation because at the end of the day, how people use this is they want to rank. They want to say, I want to knock down this gene. Can you give me a rank order of the guides I should try? And that's the sort of most prominent use case. There may be other ones, but that's sort of the real thing that people want. And so what you will want to evaluate is the rank order, actually. And you don't care about the precise value, and therefore you don't care about bias. And in general, in machine learning, no one cares at all about bias. It's the statisticians that go crazy with bias because there's a bias variance trade-off. And most machine learning people want things that work, and they know if they don't have infinite data, they'd rather have a bias and have smaller variance. So for all of you bias-obsessed statisticians. Right, so, okay, so that was the first half of the talk, and I think it gives you a sense of, how you can use machine learning. It was kind of largely off the shelf here, but the second part was we had to be a lot more creative, and I'll tell you about why in a second. Yep.
You're learning something that's not gene specific. You're going to use it.
I didn't hear.
You're learning something that's not gene specific.
Yes.
But you're going to use it. The application is someone asks, I want a guide RNA for this gene. Mm-hmm. So when you're applying this to that, what information from the gene?
Oh, okay, yeah, I see why, how that could be confusing. So the way it works is once we've learned this model, if someone goes to the server and says, I want to knock out this gene, what it does is it gets the sequence for that gene, it finds out everywhere you could apply CRISPR with that, with Cas9, for example, which is NGG, and that might be like 400 different places. Then it takes each of those 400 places and it pipes it through the model and gives the user a rank order. Yeah, we assume the sequence is known, that's right. And so, yeah, and sometimes people say, well, does the model work if, you have SNPs and these kinds of things? And yes, it does. We haven't pre-computed these kinds of things because that's impossible, but there's no reason this model wouldn't work for any sort of SNP issue as well.
So because all the examples are kind of handpicked to be pretty good,
yeah.
Is there any worry that the model would not know that the bad ones were bad? Or
sorry, they were picked to be pretty good in the, so I don't know all the design that went behind it. The one part of the design I know is they were picked to be pretty good in the sense they didn't have a lot of off-target activity. But I'm modeling on-target activity, and I'm not sure how much they could have even put in bias with respect to that because I don't think they knew very much. So, and also what they did is they systematically covered the whole gene. So, I mean, those genes were chosen, for example.
Like for the segments that had the requisite GC
exactly.
and
they picked things that they could measure in the lab, so sulfur proteins or the other cut, and then they systematically did every single thing within that. So it's
right.
I think it's relatively unbiased. Yeah. Mm-hmm. Mm-hmm.
What is the method that you are using to combine the outputs of all this ensemble trees?
To combine what?
How did you combine the outputs of the ensemble of the trees?
So the boosting algorithm itself gives a recipe for combining them. It's part of the boosting algorithm. And it basically says add the predictions together in a way that's proportional to how accurate each of the trees was, roughly speaking. Yeah. Right, so this problem is much harder for a couple of reasons. So this is the off-target problem. And so now remember that so far, everything I told you about, there was a perfect complementarity by design, right? I had a guide and it was going to match exactly to where I wanted to cut and I would only measure there. But with the off-target, accidents can happen, right? I can target somewhere, but it might be that all along the genome, I have an almost perfect match. Maybe there's one mismatch, maybe there's two, and it might still be active. And I need to model all of these because I need to basically scan. So, right, so that's why there are two problems. I need to scan the whole genome just for one guide that I want to use. Whereas before, for on-target, I just say, how well did it work here? But for off-target, I don't say, how well did it work here? I say, oh my God, like everywhere else, did it actually like cause some sort of disruption? And I also need to account for the fact that there might be these mismatches. And so you get this combinatorial explosion that you need to model, and you have very limited data. And so those two things together made this a very, very hard problem. And at the same time, we couldn't use very sophisticated models because we had so few data. And so, right. So in the end, this is how we decided to break up the problem. So the problem is going to be that for a given guide, we want to know the off-target effects. So we've decided to use this guide that's targeting this place. And so the first thing we're going to do is we're going to sort of make a short list across the genome of things we think might be active. And so I mentioned that it could be active with some mismatches. It turns out even with up to 6 mismatches, it can be active. And so I need a pretty good shortlist, right? In a perfect world, the shortlist would be the whole genome, but I can't afford to do that. So we do something a bit smarter. And we could actually use machine learning here, but we didn't because we decided to focus on machine learning for the second 2 things, which I'll tell you about. But in the first thing, what we do is we use sort of heuristics that have been floating around in the community that had to do with how many mismatches. So we know there's activity up to 6, but we also know, for example, that the distal 3 nucleotides or 4 nucleotides away from the PAM site are not super as relevant, and maybe it doesn't matter if those have mismatches. So using some sort of ad hoc rules like this that the community had observed evidence for, we build this shortlist. And in a perfect world, we want that shortlist to be as big as possible. And once we have that shortlist, then we use machine learning. So then we build a model, or we use a model where we, for everything in the shortlist, we're going to apply that model and say, how likely was it that there was a disruption here, that there was off-target activity? Remember, this is just for one guide. So now I have potentially thousands of places that I've got a score from my machine learning model. But the way people tend to use these tools, not exclusively, but probably the main use case is how I described before, is they put in a gene and they say, tell me the best guide. And the best guide means it has high on target and low off target. But so low off target, like someone wants to just scan the guides, they want to see a single number for off target activity. But I just told you that there's stuff potentially happening everywhere. And if I stopped at 2, which is actually what most people had been doing who'd started to work on this problem, then you just have this huge list of thousands of numbers, and a user can't easily rank the guides by these thousands of numbers. So you want to actually aggregate those into something that's sensible. And so one sensible thing might be sort of what's the probability overall that I'm going to disrupt the cell? Or if you're interested in, particular parts of the genome, then you might do more nuanced things. And we can't create a tool for everyone, so we just basically tried to create an aggregate score that says, what's the overall chance of having a disruption if we use this guide over everything? Of course, to do this well, we need to do step 2 well. And so that's sort of been the focus of this work. And so, right, how do we, just to give you a sense also of the explosion here, when I'm building this short list, so this is a back of the envelope computation, but if I allow one mismatch, then genome-wide, I might get roughly 100 things that will be in my short list. If I have 2, and so on and so forth. So this explodes really, really quickly. Obviously, we can't afford, and this is just for one guide, right? And we want to do this kind of for all kinds of things. And so, again, we use this sort of a heuristic, which I grayed out here because usually I talk to people who are just interested in the computation, but so you allow kind of different kinds of PAMs, and in fact, there are many pieces of software out there that don't have machine learning in them, but are designed just to basically do a heuristic search like this. And we do something that's very much along the lines of a lot of those other systems. So, right, so now we have this short list, and now I'll tell you how do we build a model and how do we evaluate this model that does the scoring. So, right, so there had been a few previous approaches to this that were not based on machine learning, and we compare to these. The best of them was actually CFD, which was developed by John Dench and his group, and that did actually very well, but we now do better than that. So now the schematic setup for the machine learning here is slightly different than the on-target case. So in the on-target case, you may recall I had only the RNA guide here, and only the activity here. And the reason I did is that it was redundant to include the target because by design it was perfectly complementary. But now I need to tolerate mismatches, so the input needs to basically be the guide and the target as well because of the mismatch problem. And you can see already that's going to be a bit harder to model. So CFD is what John had devised, and John has this incredible intuition, and I don't actually know how he came up with it, but when we started to look at it, we realized that it was very similar to something in machine learning we would call naive Bayes, a naive Bayes classifier, and so it was actually useful for us to back interpret it in that way because we could see what assumptions he had made and then sort of generalize away from that in a principled way, and that's basically how we developed this part of the model was to do that. So I don't know if I need to go into details, but the key thing about naive Bayes is that basically it's a classifier, and what the main assumption that it makes is that the features, so these are different features for one example, is that conditioned on the class, they're independent of each other. And that's, usually when you do predictive modeling, that's not the case. It's like a very strong assumption, which is helpful when you have combinatorial explosion and things like this, if it's a suitable assumption. And kind of in these kinds of data, we almost don't have a choice but to make this kind of assumption, and we do something very similar to that. Right, so, right, so we generalize away from some of the modeling assumptions there, which is one, as I mentioned, we move away from classification to regression, and we can show that helps both on the off-target, and we also have that in the on-target paper. And I don't know why people keep insisting on like truncating their real values into 0, 1s and then building classification on it, but it's something people really like to do. The other thing is that implicitly in CFD, these features here were very, very simple. Let me just I'll get to you in 1 second. So the simples were basic. The features that CFD implicitly sort of had were the following. It just said a list of where there was a mismatch, the position number, and whether it went from like a T to a C or an A to a G or something like that. Okay.
So it's okay, some kind of logistic regression for that.
I'll get to exactly,
okay.
how we do it in a second.
Right.
I'm trying to explain sort of the concepts of why we did better, where it started from, and how we got to where we were. And so you can imagine, first of all, decoupling the position here from the letters and also using like the context of the sequence and the thermodynamics and all kinds of things. And so that is actually what we do. We don't use just those features. The other thing we can do is use very nonlinear regression model for this. And so I think that's what you were asking, Alex, is like, what is the model we actually use for these things? And we actually use boosted regression trees there again. And then the other thing we did that I haven't seen anyone do, so this is, I should say, this is all very non-standard. So this is all stuff we just devised for this problem, which is to generalize away from naive Bayes or from CFD doing these kinds of things. And the last thing we did is I said there's this key assumption in naive Bayes and likewise in CFD, which is that the features, and so that could be, for example, where there's a mismatch at what position, that they are independent condition on the class. And that's a very strong assumption, and we wanted to be able to back off that assumption a little bit, but we couldn't because there's very little data. So what we did is we ended up sort of tacking on a second layer model that mitigates that assumption a little bit, and I'll explain what I mean by that. So, right. So now what we do is we need to go from this pair to some feature vector. And we use a lot of the things that I mentioned for the on-target, and then the additional thing we need to add in is this notion of mismatches, which we encode in a few different ways. So sort of the positions where they occur, the letters they occur, and also the conjunction of those. So right now, how do we actually build a sensible model? So I mentioned that there's this fundamental problem, which is there's a combinatorial explosion issue because number of mismatches, and we have super little data here, like in the thousands kind of thing. And you saw how those the mismatches just made things explode in the genome. And we can't get anywhere near enough data to model that kind of thing. So what we do is basically we make, in a sense, this naive Bayes assumption, which is that we can break it up per mismatch. So the starting piece we're going to use in this modeling is we're going to build a model which is very good at single mismatch prediction. If there's only one mismatch, how well, or how badly is there an off-target activity here? And amazingly, what is in our model is data from just one gene, CD33, and it's actually not DNA cutting, but it's actually protein knockout. And from just the CD33, every possible mismatch, that's our training data, and we build this model here. And it turns out that it generalizes quite well. We don't know how much better we would do if we had other data because we don't have other data. Now the question is, imagine I have that model, but I want to now be able to use evaluate 2 or 3 mismatch examples. How can I sort of bootstrap into that? So one is that I can use this sort of naive Bayes assumption is if this is some sort of probability and things are class conditionally independent, I can just multiply them together. And that's effectively what CFD was doing with classification in its limited feature space. But what we're going to do is say, well, you know what? We have a very small amount of supervised labels for multiple mismatch. I don't remember now the numbers, but we have some data there. It's not enough that we can just build an arbitrary number of mismatch model, but we can use it as a sort of tack on to mitigate this assumption here. And that's basically what we do, is we use a very little amount of data to say, how should I combine these? So one option is to multiply them together according to independence, and another is something more general than that. And that's basically what we do. And so, again, this first model, this is the single mismatch model. We use boosted regression trees. And then with a very limited amount of multiple mismatch data, so in this example, there's 2 mismatches, and I want to learn a function M, then we used L1 penalized linear regression. And that's like a super, super simple model. First of all, linear regression, then it's L1 penalized, which like clamps it down even further. And that's because we have so few data here. And so this actually tells you what happens in that second step. are we mitigating that assumption of independence or not? So this is the second model where we're combining the single mismatch predictions. And the way we combine them is we basically have a weight in the linear regression for the mismatch, if there was a mismatch at 1, 2, 3, 4, all the way up to 20, and then the n in the PAM, which is the wild card before the GG. And then the other thing we include is the product of those things. So the product of those things would actually just be the conditional dependence assumption. So if we didn't want to use machine learning, we would use just this feature here. And you can see that feature plays a huge role. And basically, the importance of everything else is a way to mitigate that assumption. So if this was the only feature that was on and the others were 0, it would show that this model wasn't doing anything with these data. And the fact that it's not that shows that it is able to do something extra, and that bears out in the results when you evaluate things. Does that make sense? Any questions on this? Oh, so this is the product of, so this would be correspond to the, which product?
That's important.
Oh, sorry, number of mismatch. And, oh, so it's confusing because these are really close together. So this is the number of mismatches. Number of mismatches is actually highly predictive. It's not a bad model. And it's funny because I'm not going to say that. Okay. And then this is the sum also, which doesn't necessarily make sense. I mean, the product makes sense because we know that if we didn't want to use the second layer of machine learning, we would just use product. So we basically want to make sure it's in there so that we can recover this sort of naive Bayes version of things. And these are things we just thought might help and turned out to help. Yeah.
The blue ones are sort of, do you think that's noise or signal that they're not 0?
I'm trying to think of this. This has been such a long project. I'm trying to remember if we dug that deep into this. I'm not sure for certain, but my intuition is based on the size of these, and from what I remember is that it's not just noise. I suspect it's real, but it may be that you could do pretty well without it. Yeah. So, right. This is taking actually longer than I expected. So how do we evaluate this? Maybe I won't go into all the details of the data that we use to evaluate it in the interest of time, but I'll just say that there's something very difficult about how to evaluate this problem that's not the case for the on-target. And the difficulty lies in the fact that there's an inherent asymmetry in the kinds of errors that you make. So imagine you want to use this model, and my model tells you that over here there's no off-target activity, but really there is. So that's like a really bad kind of error because I'm going to go use that guide and there's going to be an off-target activity. And if this is, therapeutic, you could kill someone. If it's just a screening thing, you'll add a lot of noise. And the flip side is that if my model says over here there is off-target activity, but there actually isn't, then I'm just not going to use that guide. And so it just constrains me a bit in how I do my experiments, but the result is not nearly as grievous. And the trade-off between these two things is domain specific. So if I'm in therapeutics, I better make sure about the off-targets. And if I'm doing drug screening, I may care less and may be able to tolerate more. And so it's not, we know there's an asymmetry, but we don't know exactly where we lie. And so what we're going to do is instead evaluate things with this newly devised metric, which we thought a lot about. And so what it is, it's based on a Spearman correlation again. And on the very right-hand side, and the way, just for visualization, it turns out to be easier to compare. CC-top is another model, and we basically evaluate how much better everything is than CC-top. Things are a bit clearer to see. And on the right-hand side here, we're going to use an unweighted Spearman. So this correlates. So this basically says our new model elevation does this much better than the baseline according to an unweighted Spearman correlation, which is the same measure I was using for the on-target. And what happens is, as I walk towards the left, on the very left-hand side here, what I do is I use a weighted Spearman where each example is weighted by the true activity. And so what that means is if an example, like a particular guide and off-target, is not active, it falls out of the evaluation. So that kind of captures an extreme version of the asymmetry that I mentioned, right? It says that if it's not active, I don't even care about it. But the thing is, you can turn this knob and gracefully go back to the other one. And so we get everywhere in the spectrum between those two things. And what's nice, so there's no theoretical reason to see this, but you see that one model almost always dominates another model. And so the beauty is that for practical reasons, it doesn't matter where you are in that trade-off, because this model, the red one's always better than the yellow, and the yellow's always better than the blue, and this kind of thing. And so that ended up being a really nice way for us to evaluate these things. These are, I don't know when these became such bad resolution, and so we do this on a few different data sets with cross-validation and things like this. And so this is just very quickly, you can see here, if you use classification, how much more poorly you do than if you start to use different versions of regression. So and this purple line is exactly the same model and features as this brown, or what is that, whatever orangey line, except the only difference is we've moved to classification. So you see there's a huge hit there. Right, so now let me just quickly go to the aggregation. So now what I've told you is for a given guide, I'm going to build a short list. I'm going to get some probability or some score of off-target activity at everything on that short list. But now I want to aggregate those into a single number using machine learning. So, and people had actually, I think there was one or two aggregate things out there, but people hadn't really evaluated them. In some cases, some of the most popular servers were out there and people were using them, and no one had literally ever evaluated the aggregate score. So we wanted to also make sure to do that quite carefully. So what we're going to do is there's going to be some particular intended target sequence, and we're going to build a short list of potential off targets, and this could be thousands of things. For each of those, I'm going to apply the two-layer model that I just told you about. So that's it tells you how to combine each of the single mismatch scores into an overall score. So this 0.43, for example, says, if this was my intended target and I design a guide for that, what's the sort of probability that I'll disrupt this region over here? It's 0.43. And then I get that for everything in my short list. And what I need to do is that's going to give me some distribution of scores. And remember, for each guide, there's going to be a different number of these. So they don't sort of map to each other. And so I need to look at this as a distribution. And to do machine learning, I need to sort of featureize the distribution. And that's how I'm going to do machine learning there. And so the sort of features of this distribution we use to get a final score is we use like the order statistics, the minimum, the maximum. And one thing that turns out to be very important, unsurprisingly, is whether it lies in a genic region or not. So that's how we compute this final score. But one thing you might be asking is, okay, well, what's the supervised data you use here? Like, how can you even get a handle on that? And so again, luckily for us, people much smarter than us in biology, and John said, hey, you know what you should look at is this viability screen. And so they do these viability screens where they target genes and then see how well with CRISPR and knock it out and see how well the cell survives. And what's known is that a handful of genes are non-essential. I don't know if it's a handful, actually, I forget the number, but there's some, and this is fairly well annotated. So you can go along the genome, you can pick out the non-essential genes. And by definition, a non-essential gene is a gene where if you knock it out, the cell survives. And so why is that important? So imagine I take a non-essential gene and I target it with CRISPR. And so that gene is, and imagine I'm successful with the on-target, or I'm not. Either way, the cell survives. But imagine that it has off-target activity. The more off-target activity it has, the more likely it is that it will kill the cell or be non-viable. So this is a proxy to overall off-target. It may not be the exact aggregate we want. I don't know what the exact aggregate we want is. I don't know. And if I had one, it would probably be different for each person in the room who wanted to use it. But I think we can all agree that that's a pretty reasonable proxy, and people have since then been leveraging this for this purpose as well. And so that's the supervised label we use. And you can see here that we compared to CFD, which is John's, and this is from this server, I should actually say, has been shut down. The last revision, we noticed that we couldn't access it anymore. They basically said, we know other people are doing, a good job on this problem, and we're going to retire the service, but it's still in the paper. And you can see that it was not well tested, and it's not performing super well, and that our model is performing much, much better than both of the other ones. But you can see actually that the correlation is very low, right? It's only 10%. So that means there's a lot of work still to be done here, and I think probably the bulk of that is actually just getting more data, because there's only so much you can do for this very difficult problem with these very limited amount of data. And so now this will be live very soon, since the paper has finally been accepted after years. And so what we now have done is we've combined our on-target and off-target modeling into a front-end web page where we've actually pre-computed for the whole human genome, anywhere you might want to, where there's a gene, where you might want to deploy CRISPR and do knockout, it has pre-computed the on and off target. So the on target's not a big deal because that's just one number, but that off target, that means for every single place, we've scanned the whole genome, we've built a short list, we've scored every part, and we've put it in. So Okay, so first of all, who can guess how many cores we had access to do this? How many would you have here?
Depends how much money you have. We use the cloud.
I see. Okay, so we use 17,000 cores. And now who can guess how long we ran that 24 hours a day? Come on, one guess. Oh my God, 6 months, no. Actually, only three weeks. They wouldn't have let us do it for 6 months. But during the course of revision, we did do it 3 times. And so that was kind of, I don't know how many places you can do that, and that was kind of a lot of fun. And when I was interviewing on the academic circuit, and I would tell people this during my interview talk, they would kind of freak out about how they were going to get me those kind of resources. So anyway, so this will be live soon, and you can put in a transcript, a gene name, and then it will basically give you, and you can sort by different things depending on what you care about. One thing we don't do that some people say is like, well, like just rank them by some overall quality, but you can't. I mean, for every person has some different amount they care about the on-target versus the off-target, and so you can just dump this all into Excel and do whatever you want with it. And so I think I'll mention this after, if people care, after people leave, but just let me finish up since time is running out. So this is very much joint work with my really, really wonderful collaborators who Nicolo was here last week, and John here at the Broad, as well as a whole bunch of other people who are at the Broad or have been at the Broad down here. And also for the last couple of revisions, we've enlisted the help of Ben and Keith and Alex, who are actually the developers of many of the state-of-the-art techniques to measure off-target activities. So the assay they developed that we originally actually have used in this paper is called GuideSeq, and we actually generated some new data to validate the model with their help. And so, right, thanks very much.